I'm Sam Sheppard. I've recently returned to Oxford, having worked here as a postdoc some years ago. I'm now professor in the Department of Biology and a member of the Ineos Oxford Institute. And my particular interest is in microbial genomics and bacterial evolution. I run a research laboratory and we sample bacteria, especially pathogens, bacteria that cause disease, from lots of different sources. So this could be from patients in a hospital that have infections, or from animals on a farm that harbour different organisms. And the typical model for our research is that we extract the DNA from those organisms and sequence it, and then we compare the genetic code to identify the genes that underlie important characteristics. For example, a gene that might make one bacterial strain virulent and another one not, or a gene that confers antimicrobial resistance, or we might be interested in genes that help bacteria to live in different places, so maybe colonize different hosts. And we bring that information together to try and get a full understanding of the genetic basis of this extraordinary variation that we see in the world around us. Understanding bacterial evolution and the emergence of traits like antimicrobial resistance has really improved as technology has got better and we've been able to sequence more genomes. So we can understand the genes and look at them and find them in all these different organisms. But that's kind of shifted the challenges, actually, from traditional laboratory microbiology, which I trained in, where we maybe would look at relatively few strains and characterize them in great detail, to looking at many, many thousands of strains. And there, we now have to think a lot more about how we're going to handle this data cascade, this big data, So a lot of my work now is around using bioinformatics. So this is science within the computer. So we will develop different analysis pipelines and different ways of comparing genomes to understand antimicrobial resistance and other important traits. And if we can understand the genes that underlie these transitions and understand the genes that are associated with antimicrobial resistance, then we can potentially intervene in the right place to stop these transmission events.